It's very nice to see many people here today. I'd like to thank you all for coming and also thank the Quebec Pain Research Network for supporting this project, as well as so many other exciting and interesting projects within the network. The project today that I will talk about is a collaboration between myself, Reza, and Drs. Lisbeth Haglund and Francis Boudre from University of Montreal, and there are several students involved as well. So we're interested in the role of intervertebral disc degeneration in driving back pain. And we know that back pain has many different causes, and disc degeneration is clearly associated with back pain in at least some individuals. The intervertebral discs are located in between vertebral bones in the spine. So this is a disc here in this picture, adjacent to the spinal cord, which is bathed in spinal fluid. The discs, this is a picture of a healthy disc that would have kind of a jelly-like goo in the middle, surrounded by layers of cartilage that hold the goo in, and it has a nice kind of squishy shock-absorbing quality. A healthy disc will have very little to no nerve fibers growing into the disc, so they're very lightly innervated, if at all. This is some of the things that happen in a degenerating disc. There are structural problems. For example, this disc has herniated and is compressing this dorsal ganglia. So that's definitely a potential source of pain. The components within the disc, there we go. The components within degenerating discs also become increasingly nociceptive and pro-inflammatory. So we have kind of a perfect storm of this becoming increasingly noxious and more likely to come in contact with sensory neurons, or finally the invading nerve fibers. So the third thing that happens is that there's more nerve fibers growing into discs, they grow in deeper. So now we have this noxious goo in contact with innervation fibers in the disc, as well as leaking into the surrounding area. So we've known for a while that degenerating discs are increasingly nociceptive when the contents contact nociceptors. But what we don't really know is what components within the disc are the ones that are driving activation of sensory neurons that produce the pain sensation. So that is essentially the goal of this project. So we're asking, the goal of the project is to ask the question, what are the components, first in degenerating discs obtained from humans with back pain, and in spinal fluid obtained from humans with back pain? What are the components that directly activate and sensitize nociceptors, thus driving pain? The ultimate goal would be to identify those and then block them therapeutically. So we have shown, this is work primarily driven by Dr. Haglund's lab, we've shown previously that human degenerating discs from individuals with chronic pain release an increased amount of many pro-inflammatory and pro-nociceptive cytokines. This is just a figure demonstrating that from a previous study. I have also collected over the years spinal fluid from individuals with and without back pain. And some of which have then had surgery for disc degeneration. In each case, we did a lumbar MRI so that we know who had disc degeneration and who had pain. And it's actually pretty common as we age for discs to degenerate, even if you don't have pain. So that allows us to separate our samples into these 3 distinct groups: individuals with pain and disc degeneration, no pain, no disc degeneration, and no pain, but some disc degeneration. And the bottom line here is that in the individuals with disc degeneration and pain, there is an increase in pro-inflammatory cytokines and other inflammatory factors in these individuals with pain. So how do we figure out what the factors are that are important in driving increased nociception? So this is our research plan, kind of in a nutshell. We will use materials collected, as I've already shown you, from painful degenerating discs and from spinal fluid obtained from individuals with back pain and those who do not have back pain. And these are the components. Mostly this is Dr. Haglin's component. This is my component. Dr. Reza Sharif is primarily driving the middle section here, which is the development of a high throughput assay system where we can take these materials from patients and screen them on nociceptors to see what components activate the nociceptors. So once, because it's very difficult to obtain primary actual sensory neurons from humans, he has developed a system where he can make nociceptive-like cells, which we call inocs here, from pluripotent stem cells. And I'll show you a little bit more about that in a moment. Once these cells are made, they can be plated on these multi-electrode arrays so that we can screen lots of different compounds all at the same time. So that was this was a big part. Developing and testing the system was a big part of the original objectives. We were then hoping to isolate and identify some of the active compounds and eventually to test them in mouse models to see if there's a therapeutic benefit to targeting them. So to develop the nociceptors, it's the induced nociceptive-like cells, you might say. The protocol is as follows. So it starts with pluripotent human stem cells, and they're kind of globular round things at first. So they're not neuron-like at all. We culture them in the presence of growth factors and transcription factors that shift them towards a sensory neuron phenotype. And quite a bit of work has been done by Reza and others at other institutions to kind of optimize what's in this mix that drives towards the phenotype. So you can see after 2 weeks, they're really starting to look like neurons. They have processes here. And typically the cells are tested at about a month, where they, by that point, if you test them, they generate these beautiful action potentials that look a lot like the responses in human sensory neurons. And they have similar resting membrane potentials to human primary sensory neurons. In addition, they express many of the receptors that are associated with nociceptors, like ATP receptors and the capsaicin receptor, and are sensitive to these mediators. So it's a pretty good model of to use for screening purposes. I skipped this important. This is probably the most important slide. So the idea was to take these 96 well plates in this system that is available at UDM and fill these plates with these little induced nociceptors And then there are electrodes all over the place. So we can do get a lot of data at once from many neurons. And the expectation and why this method was selected was because we expected it to be highly reproducible. We expected it to be easy to use. And there was some really exciting preliminary data that inspired us to go forward. Unfortunately, the reality of it was that when we tested the system in the presence of spinal fluid as well as in the absence, there was the results were not very compelling. And the big problem was that there's so much variability, even in the untreated cells, that getting meaningful comparisons to cells exposed to the pathological CSF, for example, would be nearly impossible. So that is disappointing. But that's how research goes, right? It gets us closer to the eventual solution by having excluded this one. So the plan moving forward is something just completely brilliant that Reza is starting to build in his lab. And I'm so excited about, I hope that he continues to want to collaborate with me in this area. So what he's doing is he's building a system that is completely optical. So using calcium sensitive dyes and voltage sensitive dyes, the induced human nociceptors can be developed through the protocol that I showed, and then activity can be imaged using these dyes. If the cells are transfected, either with a viral vector or the CRISPR technology could be used to make it so that the cells express light-sensitive ion channels, then we can stimulate the cells with light, and then the ion channels will open and there will be activities. So that's kind of the model. And then of course we want to use this model to do further work, eventually to do the isolations we'd hoped to do, identify the active compounds, and to do this we will probably need NIH funding or CIHR funding. We actually did apply about a year ago to NIH for this project, and we got really good feedback, but we did not make the funding cut. So this is an example of just from the literature of what the responses would look like. So you stimulate the cells with the blue light, and then you get an action potential. And what we anticipate, although the experiment will need to be done, of course, is that there will be increased spiking of greater intensity in cells that have been exposed to CSF from patients or to the disc contents obtained from surgical patients. And with that, thank you very much. I'd like to thank Ulia and Reza for their assistance developing these slides and translating them so everyone could understand. Thank you.